Phylogeny is simply the evolutionary tree of life. So it's like a family tree, but it represents how species are related to each other. So species that are more closely related to each other are closer on the tree than species that are more distantly related from each other. And that's all a phylogeny is.
Well, you could use structural data, then morphological data, and most importantly nowadays, molecular data. In terms of molecular data, you use DNA data, and also you can use RNA, and then there's a whole lot of chemical data from metabolomics, where you're looking at enzymes and also different chemical compounds that species produce in looking at their similarity with each other.
Well, they're getting better all the time. So the more data, one of the big things we've had is a revolution in the speed with which we can sequence DNA. We can now do it so rapidly. We have so much data. And because we've got so much data, we're in a much better position now to be able to estimate phylogenetic and evolutionary relationships.
Well, yes. So I think the reason for different phylogenies for the same clade is the amount of data that you use and the amount of species that you use to construct the phylogeny. So in phylogeny, the theory is more the better. The more the sequences you have, the more you can see the relationship clearly. And the more the sequences that you analyze, then it's going to be more clear, and it's going to be a solid phylogenetic representation of that particular clade. And furthermore, there are different the data analysis methods where you generate these phylogenies. So depending on these methods as well, and depending on how you analyze the particular data, then again the phylogenies are going to differ. So I think in order to generate the most robust phylogeny, what you need to have is a large data set and a good representation of whatever the species that you're going to build a clade for, in order to make it robust and a solid phylogeny. But again, it's also changing. Like if you consider the phylogeny system for the angiosperms, like every year you get an update for the phylogenetic system based on whatever the new species that you're adding, as well as the number of sequences that you're sequencing for the whole plant kingdom.
The complexities are knowing how to analyze the data. So you might have huge amounts of data, loads of gene sequences, but then what are you going to say about how those evolve in order to be able to construct the phylogeny? You've got to make some assumptions about the rates at which DNA changes, what parts of the DNA are more likely to change than other parts, because some genes evolve more rapidly than other genes, what individual nucleotides are more likely to change than other individual nucleotides, because you get different rates according to different positions in the nucleotide sequence. It starts to become a really complicated thing when you've got to consider all of these different aspects mathematically, and so you can get very different answers with the same data even, just because of the statistical assumptions you're choosing. So you can end up producing completely different evolutionary trees, completely different phylogenies, according to what assumptions you use in the first place about how DNA is evolving. So there's different assumptions you can make and different ways of analysing the data. One is just parsimony, which makes the assumption that the least amount of change is the most likely amount of change, and therefore species which show the least amount of change are most likely to be the most closely related species. But the problem with parsimony is that it isn't always the case. We sometimes see species that are quite distinct related that actually are very similar to each other. So we start to incorporate other kind of more complex mathematical models, things that involve things like maximum likelihood or something called Bayesian statistics. It's always developing this field. And what we're hoping is that by developing the mathematical models more and more, we get at a better resolution of phylogeny.
Well, I think both's important. Like it's I think it's an amalgamation of both. You have to have molecule and the structural or the morphological information in a side, and even if you can add chemical data into that, the chemotaxonomic information into that, the more the data, more the resolution, as I mentioned earlier. So I think when you can't resolve the phylogeny in terms of the chemical data or the structural morphological data, that's where the molecular data come into play. Then you can have explanation through the molecular history of those organisms in terms of resolving the phylogeny. So I think both have a equal weight in developing a phylogeny.
Molecular data, once it became really easily available, which it is now, transformed our ability to do phylogenetics, construct phylogenies, in that you just had so much more data. When you were looking at structural, morphological data, there was only a limited number of characteristics you could look at, maybe a few hundred. Now you can generate genetic sequences with tens or hundreds of thousands of base pairs. So it's so much more data that we've got to work with now. So that has revolutionized what we can do in terms of reconstructing phylogeny. But one consequence of it is that we've ended up identifying evolutionary relationships which we think or didn't realize were there before. For example, we now know that elephants are closely related to things called hyraxes, which are little furry mammals. And we would never have necessarily thought that those two were closely related prior to there being molecular data. So we start to see surprising things coming out of what the genes are telling us compared to what structural data tells us.